All right, last video, I finished up talking about muscle fibers and how each muscle fiber is a cell, and these cells are made of these individual myofibrils, which are the thick and thin filaments made of actin and myosin. Remember, thick filaments are myosin, thin filaments are actin, and then these things form this, or these filaments form this repeating units called sarcomeres of light bands and dark bands. Okay, so that's where we're picking up, and we're going to go into what the heck this picture is right here. So keep this picture in mind, or you can even have it open on your presentation as I'm talking. So these stripes form our striation pattern on our muscle fiber. So the first thing we have is the I band. I is the light band. So light has I in it. It is the light band made of just actin filaments. So there's only actin within the I band. In the center of the I band, I have something called the Z line, or it might even be called the Z disc. This holds the I bands together, and the Z disc marks the end of the sarcomere. So a sarcomere goes from Z line to Z line, and it's right in the middle of the I band. The A band is the dark band. So A, dark has A in it. This is where it appears dark, obviously, and we have actin and myosin that overlap with each other. And this is a pretty wide band, and I'm going to show you a picture here in a minute. In the center of the A band, or the dark band, there's something called the H zone. This is where I only have myosin. So it still looks pretty dark, but the shade is going to change a little bit because I don't have myosin and actin overlapping. It's just a region of myosin only. In the center of the A band, there's something called the M line. This is right in the middle of the sarcomere. Have these really big proteins that hold everything in place. Now, going back to the Z line also helps to hold myosin in place, and it does this with this protein called titin, T-I-T-I-N. It attaches to the Z disc, and it kind of, what we're going to see is this thing called the sliding filament model, where everything slides past one another. So these Z lines are going to move towards each other based on how they're connected to the actin and the myosin. And like I said before, Z line is the sarcomere. The thick filaments are made of myosin molecules, like I mentioned in my previous video, and there's two of these protein strands twisted together. But they have these things on them called cross bridges. These cross bridges, I always think of them as like golf club heads. They stick out towards the actin, and they project out from the center of the thick filaments. And these are really going to be the main things that contract our muscles. So these thick filaments and the cross bridges. The thin filaments have a couple strands of actin that are twisted around. So what we're going to see in a later video is that the cross bridges attach to the actin and pull the actin towards the center. Well, on the thin filaments, we have two things here. We have troponin and tropomyosin. Let's start with the tropomyosin first, although it's below. The tropomyosin is this protein that wraps around the actin molecules, and it's in the grooves of those little pearl-like structures. Okay, the troponin is what holds the tropomyosin in place. So troponin holds the tropomyosin in place. When we go to contract our muscle, I have to shift the tropomyosin out of position. So that whole thing is called the troponin-tropomyosin complex. When we get to the actual contraction part of the muscle, I have to move those things in order for the cross bridges of the myosin to attach, okay? So again, thick filaments, myosin, where we have the golf club head sticking out. The actin is the thin filaments, and they have troponin and tropomyosin on them. So this kind of wraps up everything you've seen here. Notice I have the I band, which is the light band, looking down at the bottom picture. Notice it's a little bit lighter, and there's just actin there. The A band is the dark band. That's where you see actin and myosin overlapping. In the center, you have the H zone. That's where I only have myosin, but what we're going to notice is that the H zone is going to disappear later. We'll see that. Sarcomere goes from Z disc to Z disc, and this is what allows us to contract our muscle. A little bit better picture, just zoomed in on one sarcomere here, really breaks it down and shows you. Notice on the myosin on this picture, you see those little cross bridges sticking out. Those are the things that are going to attach to the actin and pull everything towards the center. Also notice, and it's only labeled on here one time, but he has titin, the picture shows titin attaching the myosin to the Z disc on the left-hand side there. Something happened with my computer here. Okay, here we go. Moving on. So now we're going to structures within the muscle cell. First one we have is the sarcoplasmic reticulum, similar to the endoplasmic reticulum that you probably learned about in biology. Tubules and sacs, and this sarcoplasmic reticulum is going to contain calcium. Calcium is extremely important, specifically positive calcium ions. So sarcoplasmic reticulum is pumped in there, stored in the sarcoplasmic reticulum, and then it's going to pump it out when we want to contract our muscles. Transverse tubules, if you're looking at the picture on the right, notice there's little holes in the sarcolemma, and then those holes feed into this tube which goes deeper inside the muscle. That is a T tubule. It says it extends into the sarcoplasm and contains this fluid that allows an impulse to travel through it, an electrical impulse. So keep that in mind. The transverse tubules are extremely important because they're going to allow that impulse for contracting a muscle to get deep into the muscle fibers. Now, the transverse tubules or T tubules are between these two things of the sarcoplasmic reticulum called the cisternae. So I don't have my pointer. I'm not doing this in class, but we could talk about it in class. But notice how I have two cisternae surrounding a T tubule. Okay, so those three things make up what they call the triad. So I have a T tubule surrounded by cisternae. The cisternae are part of the sarcoplasmic reticulum. Okay, yeah, that's it there. And I'll stop that there, and we'll pick that up in the next video.